Our next keynote speaker is Professor Stéphane Avril. I apologize in advance for what I'm about to do to the French language, but please bear with me. Stéphane is the Professeur de Classe Exceptionnelle, Mines Saint-Etienne, Institut Mines Telecom in France, where he's the director of SAINBIOSE, an INSERM endorsed laboratory with over 150 researchers. His research group and SAINBIOSE are focused on cardiovascular dysfunction, where they're developing state-of-the-art computational models and applying them to cardiovascular medicine to assist physicians and surgeons in clinical treatment. Stéphane's work has received many awards and distinctions, including a European Research Council Consolidator grant in 2015 and a European Research Council Proof of Concept grant in 2021. This year, he was again awarded a European Research Council grant, this time in the advanced category, to carry out frontier research that aims to minimize the impact of arterial aging by developing better understanding of biomechanical forces exerted on cells. Please welcome Professor Stéphane Avril to the stage.
Thanks for very kind introduction, and I feel very honored to be here and also very humbled. I've known Galway forever since I started my career. I was always admirative of what's going on here. With Peter McHugh and other colleagues in my domain, I was always admiring. And I also feel a bit emotional because actually, when I started my career, so you see here, I come from Lyon, and I'm part of a big national institute, Institut Mines Telecom, with different campuses, and it results from the merge of School of Mines and School of Telecom in France into a big national school. And so there was this School of Mines in Saint-Etienne, and then beginning of just after the year 2000, it was decided to start a new center on new technologies, and then health science and biotechnologies was the target. And I got a position in 2007 there. I was appointed professor, and I really benefited from a lot of opportunities thanks to the launch of this new center. And I think that my talk today, I would like to especially talk to the youngest of the ones who are here, because this new institute here is going to be a lot of opportunities for the youngest. And I know how this was beneficial for my career. A lot of things I would not have been able to do them if there was not such an initiative of gathering people together in a small town in France. So I'm going to talk about in silico modeling in cardiovascular medicine. That was a request, but that's also what I do, and especially about digital twins. So I don't know if any of you have already a digital twin, because that's something maybe you've heard about, but maybe some of you already have it. It's probably not possible now to have a digital twin of your own body, of all your organs and so on, but it's possible to have some of a single organ or a system. And what the message I would like to give today is that it's becoming a product, okay, that companies are selling. And then this is a technology, okay? This is not only a topic of research, even if things are improving. And so the idea is that you have a copy of yourself, and this copy of yourself or one of your organs is going to be personalized. And then at the same time, there will be computer models solving equations of physics that, are satisfied by your blood or by your muscles or so on, so that then it's possible to make predictions. So what's going to happen if we change something to your body, for instance, if you undergo a surgery? And so then this is how you can personalize the treatment or an intervention. And so that's the idea. And this is something that is pushed a lot at the European level with different initiatives now, so that, an initiative, the name is EDIT, and tries to really put people together around virtual twins or digital twins, which usually means the same. And the concept is quite complex, actually, because you have all the data that you can acquire for a patient, and then you have all the equations and the simulation tool that you can use and to make everything linked together. And if you think about the time flow to develop a digital twin, it can be very long. And so I would not be able to teach that nowadays. And but what I can do is that I can talk of my own experience, and that's going to be simpler. So my own experience, and again, I think that really fits well with what's going on here, is that when I started to do research in biomechanics, then I benefited from this new institute, and I could sit at the same table with a surgeon. Okay, with plenty of surgeons, but especially one was really committed to do something with me, and it was posing the problems and asking me to give answers because I was a scientist and he was in the OR. Okay, so that's the beginning of my own experience, and what was really good for us is that this school of mine had a campus. But then we benefited from local investment so that we could build a new building. So you see, this is the building where I work, okay, and everything else. So it's a school of engineering where I work, but everything else is a medical school from the General University and the hospital. So it's really engineers. In the middle of other people who are more doing biology or clinicians. So that was also very interesting because we had a unique location for this research. And also that was quite a risk for our School of Engineering to put people outside of the campus and so far from the rectorate, and that we enjoyed, but also with the risk of being more independent. So that's the building. So it's a small center. We have only 4 topics where we have teams, and I'm in the soft tissue biomechanics, but we have also biomaterial engineering, inhaled nanoparticles for respiratory diseases, and also something that has been very successful related to engineering of care systems. So this is using AI for modeling the flows of hospitals and managing healthcare production in the hospitals. And my own research was always focused on one specific disease. This is aneurysms of the aorta. So the aorta is this artery that also you saw several times in Ellen's presentation. And this is the main artery that takes blood out of the heart and then distributes it to the other arteries. And then for some different reasons, it may balloon at different locations in the thoracic part or in the abdominal part. And these dilatations that occur, they are irreversible. So the main reason why they occur, it's like degeneration of the tissue for different reasons. But why it's irreversible is because the same pressure that you have in the blood, when the artery balloons, the pressure has a higher stress on the wall. And so then it cannot go back. And there's no treatment. The only solution is surgery. But surgery is not also the best, because if you do, for instance, open heart surgery for this kind of aneurysm, then it may have more risk than leaving the aneurysm in the body, because most of these aneurysms, they remain stable, and you never know which one is going to burst. So that was always the first question that the surgeon was asking to us. Could you help us with computer models to predict which one is going to burst and which one is not going to burst? That was a very difficult question, and so that's not the first one we could answer. And so then we worked on another question first, and this other question was related to helping still, if for sure that patient is going to have a surgery, can we help the surgeon to make the planning of that surgery? And especially one type of surgery, which has been developed a lot in the 1990s for these aneurysms. And this surgery is what we call endovascular procedure. So it means that instead of opening the patient and then opening the aorta and replace the balloon part, the aneurysm, by a graft, then we take a catheter, and then in this catheter we have a stent that is covered with a textile, and then you bring by femoral access and then by navigation, you bring that catheter where there is the aneurysm, you deploy it, and then it will seal and then prevent the blood pressure to apply pressure on the aneurysm again, and then the aneurysm will shrink. So that's something that is minimally invasive, but the problem is that it has to be planned very precisely, and especially the aneurysms which they cover also the renal arteries. Because if then with a stent you cover the renal arteries, then you would prevent the blood to go to the kidney. So then this would be another problem for the person. And nobody here in this room has the same location for the renal arteries. So it has to be precise. So you can measure that with imaging. But the problem is that when you deploy this stent, then they will move because the aorta is elastic. So that's the first thing we did. And we patented to predict this, the deployment with a computer model using the finite element model. And then we started to implement that in a platform that we call a digital twin, and that led to founding this startup, Predisurge, that now is proposing a service to stent manufacturers because they usually offer that service of planning to their customers, the hospitals. But this has been fully automatized thanks to these computer models. So this is how it looks like. So this is a platform that reads the preoperative scans that every patient gets before the surgery, and then download automatically. And then using artificial intelligence, it will reconstruct directly a mesh. So this is a way of discretizing all the aorta so that we can solve the mechanical equations on that. And then after, it's going to assess different interventional strategies and different position of the stents until getting the best location. And what is the criterion for the best location? So this is something that has recently been proposed. This is the Embolic Risk Index, ERI. This ERI has been validated clinically, so it enables before the surgery to predict how much risk there is that after putting that stent graft, there would still be leaks. The blood would still apply pressure on the aneurysm, and so this has been validated on more than 1,000 patients at the moment. And Predisurge is also doing other things like, for instance, predicting also possible kinks when you use a stent for the carotid artery. That is just shown here. But I would like to now move on to something else. And again, if I think about the different opportunities, so as I said at the beginning, we always wanted to go further and make some predictions on these aneurysms. Are they going to burst? But for that, we need to predict how they are going to grow using computer models. And that was kind of a difficult questions because the aneurysm growth is difficult to understand. So we could gather a lot of data on cohorts of patients thanks to the proximity with hospitals. And some of these patients, we could get both imaging from CT scans that are dynamic, gated. So then we have like we can track the motion of the wall of the aorta, and then with that motion we could relate to the pressure, and then identify the regions locally. What is the stiffness, the Young's modulus of that aneurysm, and make maps of these Young's modulus as you see here, and see that there are locations in the aneurysm that are much stiffer than other regions. And at the same time, some of these patients also, they had a 4D flow MRI. So MRI is also a way of imaging inside the body. But the 4D flow is a sequence that you can track what's moving in the body, especially the blood, and then measure velocity vectors in the blood. And so from these velocity vectors, if we look at them close to the wall of the aorta, then we can detect their gradient and relate them to the shear forces that the blood applies on the wall of the aorta. So this shear force is also a vector, and we can find that this vector vanishes at some locations. And the locations where it vanishes, you see, they correlate very well with the regions of higher stiffness. So as long as we could find that, then we could come up with a kind of mechanism that we could implement in our software. And this mechanism is that the cells that do not receive enough shear forces, then they are going to activate some pathways that induce a modification to the wall. And what modification? So we put that in a very simple mechanical model. So stress versus strain for people who do mechanics. And so here you see the curve that you would expect for the wall, for an aortic wall, where the stress increases like this, and the set point that you see here is for an average pressure applied on the aorta. And then when we have this hemodynamics that is disturbed that I was showing before, then this will induce reactions that produce more enzymes in the wall of the aorta. And then this is an injury that will induce a softening of the wall. There are less fibers, especially elastin proteins that contribute to take the stress. But then there are other cells in the wall that are called the smooth muscle cells. And these smooth muscle cells, they will relax because they have more stress. So then this is kind of stretching them too much. So there will be a reaction of production of another protein that is called collagen that will heal the wall, but with some irreversible distension, and this is what caused the aneurysm. So by putting that in a model, in a finite element model, so still the same as the one that we used to deploy the arteries, we were able to simulate the growth of aneurysms and get then these digital twins of aneurysm growth. And where you see here in red, for instance, this is the region where there has been like this process of compensating by the injury on elastin by new collagen. But you see that the aneurysm grows everywhere, because the idea is that when you change the equilibrium of forces on all the cells, then it will distribute far from the region where you start the injury. So that was interesting. And but another problem is that actually when we came to see biologists and that we had a model for aneurysm growth, then I don't know if there are many biologists in the room, but the first thing that they say, well, you can't do biology if you don't have cells, okay? So then we had to integrate biology in our models so that we could better describe what's going on in the aneurysms and also work on possible treatments. And so cells, okay, so where are the cells? So as I said, we have cells, the smooth muscle cells in the wall of the aorta. They are usually stacked, like you see in this schematic, in different layers. In a human aorta, you have between 70 and 80 of these layers. And in gray and black, you see what is called the lamella of elastin. And so you have this stacked structure. And what is really interesting is that these smooth muscle cells, actually, there are many possible genetic mutations that can induce a dysfunction of their contractility. So they are able to contract and to attach to their surrounding matrix. And there are a number of genetic diseases that have been induced, for instance, on animal models. And the first thing that you see in these animal models is that they develop an aneurysm, okay? So it's very interesting to see that if you just change something in the cytoskeleton of that cells, then an aneurysm is going to occur. So why, I mean, how things they translate from that scale to the upper scale. So that was really the question we were thinking of. And so we started to study these cells. So we took cells from we took cells from, when we were collecting tissue, so we could extract the cells, and then we tested these cells by traction force microscopy. So then we culture them in hydrogels, and then these hydrogels, they have these fluorescent beds here in green that you can see moving, and so we can relate this motion to the tension that the cells they apply. And then, thanks to that, we can see what are the forces applied by these cells, if they come from aneurysms or if they come from real patients, so that we can relate these forces to the upper scale. Okay, and this upper scale, then we know what is the force that the aneurysm, that the cell try to maintain in these aneurysms. But the other thing that we started to do that is the thing I'm going to show just in this slide afterwards, is that also we can also understand what is going to happen inside the cell when they apply these forces. And this is what is also very important. So we develop now a digital twin of the cells. And what you see is that in this digital twin, you see the cytoskeleton. So you have these fibers that are made of actin, and then there is a complex with myosin that allows these fibers to be tightened. And you see that in the middle, you have a nucleus of the cell. And actually, if we zoom on it, then when the cell is applying forces by pulling on the extracellular matrix, then it's squeezing this nucleus. And so the nucleus is really involved and has to react to the biomechanics that is applied, I mean, from the upper scale down to that scale. And we could see that, for instance, if we change the stiffness of the extracellular matrix, we change a lot the strains that we have in the nucleus. So these effects were at some point the seeds for the recent projects that we started to think about, and that relate to fighting against vascular aging. So I come back also to a thought that was given this morning. I don't know in which round table, about like the need of having like some time to think. And I have to say that I took a sabbatical during 1 year, and that was really useful to just think about other things than just wanting to cure and detect or diagnose aneurysms, and also think that this could also be useful with respect to vascular aging. So let me explain. So what I said before is that actually you have a correlation between the mechanics at the tissue level and the mechanics down to DNA gene expressions in the nucleus of the cells. And so this is what explains that there are feedback loops that any change will have consequences in the maintenance of the wall. And then we started to think that actually, what's the functional unit? What's the functional unit where everything is regulated? This is the square here that we have. So this is the functional unit is what's surrounding the smooth muscle cells, so that I'm showing here. And so we were thinking, so if we want to design anti-aging treatments, then we have to understand something. This thing is that aging is a natural process. We are all aging, and this manifests especially in our arteries by fragmentation of elastin, and this is the same effect that causes also wrinkles on our skin. We cannot really avoid that. But this effect is compensated by more and more forces taken by our smooth muscle cells, as I was explaining. And so the arteries, they stiffen, but we are still like smooth muscle cells engaged to control that. And so when we can't handle that with the smooth muscle cells for different reasons, some of them, for instance, they lose their function, then it's a tipping point after which irreversible processes start, and then we lose the function of smooth muscle cells. So then we have to find a treatment, and there are many rejuvenation treatments that are tried, but more like through clinical trials, that will sustain the smooth muscle cells before these irreversible effects. So that's why we thought that a digital twin could help to assess these effects and then try to find a way of controlling the role of smooth muscle cells before we reach this tipping point. So that's the new project that we're starting with this recent ERC Advanced. And so the idea is that before we had we had digital twins at the scale more of the tissue, and so now we are going to integrate all the cells, thousands of cells in a representative volume element, and then we are going to distribute the different populations of these cells depending on their phenotype. And so there are different phenotypes of these cells that we can assess thanks to a new technique that we acquired, that is a spatial transcriptomics imager, so that we can detect all the RNA in tissue slides and then find which gene they correspond to, and then relate that to clusters of cells that then will relate to the level of contractility that these cells they have. And so then we can put that in a computer model and see how this how it will contribute to the distribution of stresses inside the wall. So that's the new project that I wanted to mention. So with that, well, I would like to conclude. So with acknowledging the work of many people. So all this group, and the international collaborators, again, most of them, I could never have built that without them, but also without the opportunity that I was given thanks to the launch of this new center 20 years ago. And so I hope that the launch of this center here is going also to offer a lot of opportunities to the young people that are here, and that maybe in maybe 20 years, one of them is going to be invited in France for a new launch of a new center, and that we can have this cycle of research for, again, many years. Thank you for your attention.